let me ask for a show of hands. How many people here are over the age of 48? Well, there do seem to be a few. Well, congratulations, because if you look at this particular slide of U.S. life expectancy, you are now in excess of the average lifespan of somebody who was born in 1900. But look what happened in the course of that century. If you follow that curve, you'll see that it starts way down there. There's that dip there for the 1918 flu. And here we are at 2010, average life expectancy of a child born today, age 79, and we are not done yet. Now, that's the good news, but there's still a lot of work to do. So, for instance, if you ask, how many diseases do we now know the exact molecular basis? Turns out it's about 4,000, which is pretty amazing, because most of those molecular discoveries have just happened in the last little while. It's exciting to see that in terms of what we've learned, but how many of those 4,000 diseases now have treatments available? Only about 250. So we have this huge challenge, this huge gap. You would think this wouldn't be too hard, that we would simply have the ability to take this fundamental information that we're learning about how it is that basic biology teaches us about the causes of disease, and build a bridge across this yawning gap between what we've learned about basic science and its application. A bridge that would look maybe something like this, where you'd have to put together a nice shiny way to get from one side to the other. Well, wouldn't it be nice if it was that easy? Unfortunately, it's not. In reality, trying to go from fundamental knowledge to its application is more like this. There are no shiny bridges. You sort of place your bets. Maybe you've got a swimmer and a rowboat and a sailboat and a tugboat, and you set them off on their way, and the rains come and the lightning flashes, and oh my gosh, there's sharks in the water, and the swimmer gets into trouble, and uh-oh, the swimmer drowned, and the sailboat capsized, and that tugboat, well, it hit the rocks, and maybe if you're lucky, somebody gets across. Well, what does this really look like? Well, what is it to make a therapeutic anyway? What's a drug? A drug is made up of a small molecule of hydrogen, carbon, oxygen, nitrogen, and a few other atoms, all cobbled together in a shape. And it's those shapes that determine whether, in fact, that particular drug is going to hit its target. Is it going to land where it's supposed to? So look at this picture here, a lot of shapes dancing around for you. Now, what you need to do if you're trying to develop a new treatment for autism or Alzheimer's disease or cancer is to find the right shape in that mix that will ultimately provide benefit and will be safe. And when you look at what happens to that pipeline, you start out maybe with thousands, tens of thousands of compounds. You weed down through various steps that cause many of these to fail. Ultimately, maybe you can run a clinical trial with 4 or 5 of these. And if all goes well, 14 years after you started, you will get one approval. And it will cost you upwards of $1 billion for that one success. So we have to look at this pipeline the way an engineer would and say, how can we do better? And that's the main theme of what I want to say to you this morning. How can we make this go faster? How can we make it more successful? Well, let me tell you about a few examples where this has actually worked. One that has just happened in the last few months is the successful approval of a drug for cystic fibrosis. But it's taken a long time to get there. Cystic fibrosis had its molecular cause discovered in 1989. By my group working with another group in Toronto, discovering what the mutation was in a particular gene on chromosome 7. That picture you see there, here it is, that's the same kid. That's Danny Bissett. 23 years later, because this is the year, and it's also the year where Danny got married, where we have for the first time the approval by the FDA of a drug that precisely targets the defect in cystic fibrosis, based upon all this molecular understanding. That's the good news. The bad news is this drug doesn't actually treat all cases of cystic fibrosis, and it won't work for Danny, and we're still waiting for that next generation to help him. But it took 23 years to get this far. That's too long. How do we go faster? Well, one way to go faster is to take advantage of technology, and a very important technology that we depend on for all of this is the human genome. The ability to be able to look at a chromosome, to unzip it, to pull out all the DNA, and to be able to then read out the letters in that DNA code, the A, C's, G's, and T's, that are our instruction book and the instruction book for all living things. And the cost of doing this, which used to be in the hundreds of millions of dollars, has in the course of the last 10 years fallen faster than Moore's Law, down to the point where it is less than $10,000 today to have your genome sequenced or mine, and we're headed for the $1,000 genome fairly soon. Well, that's exciting. How does that play out in terms of application to a disease? I want to tell you about another disorder. This one is a disorder which is quite rare. It's called Hutchinson-Gilford progeria, and it is the most dramatic form of premature aging. Only about 1 in every 4 million kids has this disease. And in a simple way, what happens is, because of a mutation in a particular gene, a protein is made that's toxic to the cell, and it causes these individuals to age at about 7 times the normal rate. Let me show you a video of what that does to the cell. The normal cell, if you looked at it under the microscope, would have a nucleus sitting in the middle of the cell, which is nice and round and smooth in its boundaries. And it looks kind of like that. A progeria cell, on the other hand, because of this toxic protein called progerin, has these lumps and bumps in it. So what we would like to do, after discovering this back in 2003, is to come up with a way to try to correct that. Well, again, by knowing something about the molecular pathways, it was possible to pick one of those many, many compounds that might have been useful and try it out. And in an experiment done in cell culture, and shown here in a cartoon, if you take that particular compound and you add it to that cell that has progeria, and you watch to see what happened, in just 72 hours, that cell becomes, for all purposes that we can determine, almost like a normal cell. Well, that was exciting, but would it actually work in a real human being? This has led, in the space of only 4 years from the time the gene was discovered to the start of a clinical trial, to a test of that very compound. And the kids that you see here all volunteered to be part of this, 28 of them, and you can see as soon as the picture comes up that they are in fact a remarkable group of young people, all afflicted by this disease, all looking quite similar to each other. And instead of telling you more about it, I'm going to invite one of them, Sam Burns from Boston, who's here this morning, to come up on the stage and tell us about his experience as a child affected with progeria. Sam is 15 years old. His parents, Scott Burns and Leslie Gordon, both physicians, are here with us this morning as well. Sam, please have a seat. So Sam, why don't you tell these folks what it's like being affected with this condition called progeria?
Well, progeria limits me in some ways. I cannot play sports or do physical activities, but I have been able to take interest in things that progeria luckily does not